Biology, the scientific study of life. A programmed cell death, apoptosis. Lineage analysis of C. elegans has underscored another outcome of cell signaling that is crucial in animal development. Programmed cell death, or apoptosis. The timely suicide of cells occurs exactly 131 times in the course of normal development in C. elegans at precisely the same points in the cell lineage of each worm. In worms and other species, apoptosis is triggered by signals that activate a cascade of suicide proteins in the cells destined to die. During apoptosis, a cell shrinks and becomes a lobed called blibbing. The nucleus condenses and the DNA is fragmented. Notice this figure. Back to this one, notice here. Genetic screening of C. elegans has revealed to keep up apoptosis genes SID3 and SID4. SID stands for cell death, short term, which encode proteins essential for apoptosis. The proteins are called SID3 and SID4 respectively. These and most other proteins, notice that SID3 and SID4, and this one, red one, SID9. These and most other proteins involved in apoptosis are continually present in cells, but in inactive form. Those protein activity is regulated, in this case, not transcription or translation in C. elegans, a protein in the outer mitochondria membrane called SID9. This one. The product of gene SID9 serves as a master regulatory of apoptosis, acting as a brake in the absence of a signal promoting apoptosis. When a death signal is received, notice that, is received by the cell, the apoptosis pathway activities proteases and nucleases, enzymes that can cut up the proteins and DNA of the cell. The main proteases of apoptosis are called caspases. In the nematode of Chef, caspases is SID3. In humans and other mammals, several different pathways involving about 15 different caspases can lead to apoptosis. The pathway that is used depends on the type of cell and on the particular signal that triggers apoptosis. One important pathway involves mitochondrial proteins. Apoptosis pathway proteins or other signals somehow cause the mitochondrial or outer membrane to lack, releasing proteins that promote apoptosis. Surprisingly, these include cytochrome C, which functions in mitochondrial electron transport in healthy cells. Notice this figure. Back to this one. Notice also zygote here, first cell division. Notice also this outer skin, nervous system, and germline, future gametes, and mass culture. Notice also mass culture, gonads, nervous system, outer skin, mass culture. Notice also the time after fertilization, hours. Also intestine here of closer look to intestine, eggs here. Notice that. Okay. Back to SID9 or SID3, but cuts as a cell death factor when released. Factors when released from mitochondria. The mitochondrial apoptosis of mammals uses proteins homologs to the worm proteins SID3 and SID4 and SID9. Mammalian cells make life or death decisions by somehow integrating the signals they receive, both death signals and life signals, such as growth factors. A built-in cell suicide mechanism is essential to development in all animals. The similarities between apoptosis genes in nematodes and mammals, as well as the observation of the apoptosis occurs in multicellular fungi and single-cell yeasts, indicates that the basic mechanism evolved early in animal evolution. In invertebrates, apoptosis is essential for normal development of the nervous system, for normal operation of the immune system, and for normal morphogenesis in hands and feet in humans and paws in other mammals. Notice this figure. A lower level of apoptosis in developing limbs accounts for the webbed feet of ducks and other water birds, in contrast to chickens and other land bears with non-webbed feet. In the case of humans, the failure of appropriate apoptosis can result in webbed fingers and toes. Also, researchers are investigating the possibility that certain degenerative diseases of the nervous system result from the inappropriate activation of apoptosis genes, and that some cancers result from failure of suicide cells that have suffered irreparable damage, including DNA damage that could lead to cancer, normally generate internal signals that trigger apoptosis. Studies on the roles of addiction and apoptosis during developing of C. elegans began less than 30 years ago by Sidney Barnard, John E. Solstone, and H. Herbert. The importance of their studies was highlighted in 1923 when the Nobel Prize of Medicine was awarded to these researchers for significantly advancing our understanding of how genes regulate organ growth, such as the nominated vulva and the process of programmed cell death. Back to effect of apoptosis during paw development in the mouse. Notice that in mice, human, and other mammals, as well as land birds, the embryonic region that develops into feet or hands initially has a solid plate-like structure. Apoptosis elements the cells in the interdigital regions, thus forming the digits. The embryonic mouse paw shown in these light micrographs are stained so that cells undergoing apoptosis appear bright yellow. Notice that. Apoptosis of cells begins at the margin of each interdigital region left, because as the tissue in these regions is reduced middle and is no longer visible when the interdigital tissue has been eliminated. Notice that. Okay.